First, I want to thank the Provost Office, my department, and the BSc community for enabling me to take a sabbatical last spring. Sabbaticals not only provide faculty with opportunities to delve deeper into their research, but also allows faculty opportunities for reflection and exploration. When I applied for this sabbatical, I envisioned that it would have been spent fully in the lab. However, just 1 week prior to starting, my plans changed, as plans tend to do. In late December, my colleagues and I were notified that we were awarded $1.25 million to develop a new environmental justice focused curriculum for middle schoolers in Africatown. So, while I will talk about my benchtop research and drug discovery, I also want to share with you some of the work we've done over the last year in Africatown. As I mentioned, sabbaticals often provide time and space for exploration, which for me meant an opportunity to read outside of my discipline. Over the last year, I've discovered a number of beloved authors such as T.J. Klune, Octavia Butler, Kristen Hanna, and Janine Cummins. When you immerse yourself in literature, it is hard not to find concurrent themes that reflect your circumstances or give you pause. For me, that theme was the impact of words and the importance of voice, compassion, and resiliency. Because while words can be weaponized to shame people into silence, they can also be crafted with compassion to elevate the voices of everyone around you. One such book was The Invisible Life of Addie LaRue by Victoria Schwab. And while our protagonist Addie isn't actually invisible, she is utterly forgettable, and not in a figurative way. She is literally forgettable. This book opens to Addie as a young woman in 1700 France running away on the eve of her prearranged wedding. We find Addie so desperate to control her own destiny that she makes a deal with the devil. In exchange for her soul, she asks the devil to allow her to live a life of her own choosing, and he agrees to the deal. The next morning, Addie awakens to realize no one remembers who she is, not her family or her neighbors, no one. And while she can interact with people and have conversations, she is unable to leave a lasting impression on that person's memory. As soon as people lose sight of her or fall asleep, they forget her. If she tries to write something down, it disappears. Anything she tries to break fixes itself. Even her footprints disappear. Addie quickly realizes that the devil deceived her, that while he had given her self-agency and control of her destiny, he had stolen her voice and her ability to make an impact on the lives around her, all to shame her. Then, to add insult to injury, he also made her immortal. However, as we continue to read, we find that one of the underlying themes of the book is that the culmination of little things can have a big impact. And Addie, in order to make meaning from her life, would have to work exceedingly hard to influence the work and ideas of other people. Throughout her existence, Addie would spend months with an artist, introducing herself repeatedly, spending the day with the person over and over again until her message became so embedded within their own thoughts that they would unconsciously compose her song, write her novel, or paint her vision. And while she did eventually find a way to make an impact through the voice of others, it was never directly attributed to her, and her influence was always forgotten. Through her novel, Schwab teaches us simultaneously to never underestimate the importance of voice to influence the world around us and about the resiliency of people demanding to be heard. As a chemist, I certainly have grown to appreciate the impact of things we often take for granted. Our research revolves around understanding how the smallest particles come together to shape and influence our lives, things we cannot see or measure directly. We instead rely on measuring their impact. We all got a crash course on the big impact of small things in 2020 when a virus that is 1/10 the size of a pinhead stopped the world in a matter of a few weeks. It is through the COVID-19 pandemic that the modern world got reacquainted with just how intimately our economy, culture, politics, and technology are entangled with infectious diseases. And just as Addie LaRue teaches us, the cure for such infectious diseases only comes from the cumulative voices of lots of people doing their part. For my research group, our goal is to identify and describe the tiny pieces of machinery within an infectious microorganism that, if stopped, could wreck the invasive organism. The challenge, though, is to ensure that that piece of machinery does not resemble our own, so that when we stop it, it only kills the infection and not us. That is why the spike protein of COVID-19 was so attractive to biochemists. It was unique to the virus and plays a major role in the infection pathway. For big pharma, the first step in identifying a drug target is to first identify a disease you want to investigate. For most, that involves weighing the potential profitability against the research and development cost. So diseases that impact larger, insured populations are often chosen. One of the benefits of working in academia, however, is the freedom to give voice to neglected diseases or diseases that impact poorer, underserved populations. Grant funding directed by the government can also influence the decisions of both academic researchers and pharmaceutical companies, as grants can help offset the cost burden. Developing novel therapeutics for neglected diseases is half the battle, though. Often there are effective treatments readily available in developed parts of the world. However, for our poorest, most underserved populations, these treatments are out of reach physically, if not financially. Coupled this with immunocompromised populations such as the elderly, young, or those co-infected with HIV, then something as benign as the diarrheal disease cryptosporidosis can be the leading cause of illness and death for our poorest communities in the world. If you are not familiar with cryptosporidosis, it is an illness caused by a waterborne parasite that, when ingested from contaminated water or food, can cause gastrointestinal illness. For healthy individuals, this illness is typically a nuisance characterized by diarrhea and stomach aches. For those with weakened immune systems and or limited access to clean water, it can be lethal due to dehydration. The microorganism that causes the illness, C. parvum, is a hardy parasite that can lay dormant and withstand extreme fluctuations in temperature. However, they are particularly happy in warmer waters and are resistant to chlorination. To date, there is only one FDA-approved treatment on the market, but it is not approved for immunodeficient patients. At a retail cost ranging from $300 to $1,000 per dose, clinics that serve under or uninsured communities often treat the symptoms rather than prescribe the broad anti-infectious drug Nitasafe. For most of us in the United States, we only take notice when C. parvum outbreaks cause water parks to shut down. But for underserved communities, it can wipe out the main supply of drinking water for both humans and livestock. Indeed, in the poorest parts of our world, one of the leading causes of death for children 5 and under is cryptosporidosis. For children who do survive infection, there is still the potential for long-term side effects, such as reduced cognitive development and cardiovascular disease due to damage in the endothelial tissue. Over the last few years, we have been working with my students and my collaborator, Dr. Deveshish Chattopadhyay from UAB, to better characterize a potential new drug target for C. parvum, the enzyme pyruvate kinase. For those of you in the audience who have taken biochemistry or cellular biology, then you know that pyruvate kinase is an essential enzyme for cellular function in nearly all organisms. Thus, targeting and stopping pyruvate kinase could then effectively kill C. parvum. Pyruvate kinase is the enzyme that governs the last control step of glycolysis, or the process in which sugar is broken down to form energy in the cell. As seen here, pyruvate kinase catalyzes the conversion of phosphoenolpyruvate to pyruvate while producing a molecule of ATP, the energy currency of our cells. Pyruvate is a key metabolite in the cell and intersects with many physiological processes. It can be converted into acetyl-CoA for fat synthesis or used by the citric acid cycle to make more energy. It can be turned into lactate and transported throughout the body. It can be used in protein synthesis, or in the case of yeast cells, it can be used to make ethanol. Therefore, controlling the rate at which pyruvate is made by controlling the enzyme pyruvate kinase can directly impact nearly all of the metabolic processes of an organism, and stopping it altogether would be lethal. The problem is that our cells also have this same enzyme, and if we design a drug that isn't specific for the pyruvate kinase of C. parvum, we could kill both the microorganism and the host. So the job of my research team is to discover the differences between C. parvum pyruvate kinase and human pyruvate kinase. First, we looked at the amino acid sequence for CPK and compared it to other species. Here, we wanted to assess where there were differences. Pyruvate kinase is considered a highly conserved protein, meaning the amino acid sequence of PKs found in our cells is very similar to those found in other species. Therefore, differences found in one species compared to the other is often evolutionarily important in the survival of that specific species. And indeed, when we look at CPK versus other species, we found something unique. Two unique cysteine residues that are far apart in the linear sequence of the peptide chain. Now, cysteines are special amino acids in that if you get two of them close enough together in space, they can form a strong bond and are often found to stabilize the three-dimensional structure of a protein. Indeed, when we solved the crystal structure of this protein, we did discover that these residues form a strong linkage in the three-dimensional structure, holding the overall tetrameric structure as a pair of domains, creating what we call a dimer of dimers. Typically, pyruvate kinases exist as a tetramer of monomers, meaning 4 copies of the pyruvate kinase chain fold and assemble together using non-covalent interactions. Non-covalent interactions are like the pull of bringing two magnets together. If close in space, they will adhere to each other, but it doesn't take much force to pull them apart either. A covalent linkage from a disulfide bridge between two cysteines, however, can keep the domains together regardless of how hard you try to pull them apart. To our knowledge, the pyruvate kinase of C. parvum is the only known construct that has this property, thus making it highly unique and is likely highly important in the functionality of the protein. So, to better understand their role in the functionality, we created a new construct of CPK that we called the double mutant by changing two cysteines to a different amino acid and compared its structure, stability, and kinetic properties to the original or wild-type construct. To honor this audience and allow time to discuss my other research focus, I won't go into detail of our experimental methodology, but will walk you through the analysis of our results and summarize our findings to date. First, we determined that the disulfide bridges do play a significant role in the stability of the molecule. For one, we had an exceptionally difficult time getting a stable crystal of the mutant molecule that would allow us to study its structure through X-ray crystallography. This finding was supported by our thermal denaturation studies, where the double mutant demonstrated to have two melting temperatures, as shown by the two major peaks at approximately 35 and 60 degrees Celsius in the thermogram, whereas the wild-type construct shows one major peak of interest at 65 Celsius. Now, the presence of the two peaks in a thermogram such as this can be explained in two different ways. First, as depicted in the example to the left, where a protein has two unfolding events. One where there is a dissociation of major subunits to create a partially unfolded structure at the lower temperature, followed by a complete unfolding event at the higher temperature. The other possibility is that we actually have two structures, one that is less stable and unfolds at a lower temperature, and one that unfolds at a higher temperature. We can begin to probe the thermodynamic effect of various ligands or small molecules that bind pyruvate kinase by repeating the DSF analysis in the presence of these molecules. Often, if a molecule binds a protein, it will have a stabilizing effect on the structure and will shift our peaks. And what we discover when we look at these results is that the binding of ligands to the wild-type CPK doesn't do much to impact its thermal stability. But with a Tm near 65 to 67 degrees Celsius, there really isn't too much room for improvement here. The interesting results, however, are for the double mutant. Here we see that while these ligands may not impact the higher temperature peak, we see that they do impact the lower temperature peak. And we begin to see a trend where certain ligands or combinations of ligands will begin to push the double mutant data to converge into one peak, much like the wild-type construct. This information is important for two reasons. It not only provides insights into the potential thermodynamics of the protein structural assembly, but also provides us clues into how we might be able to obtain more 3D, more stable crystals for 3D protein structure elucidation of the double mutant. Indeed, we are already starting to see promising results when we try to grow crystals in the presence of PEP, G6P, and AMP, and hope to obtain a quality crystal for diffraction soon. While the impact of the disulfide bridge on the overall stability of the enzyme was not surprising, what was surprising was how the differences we saw in how the ligands affected the thermal stability and potentially the enzyme structure of the mutated form, but not the wild type. This effect suggests to me that the two peaks in the thermogram represent an equilibrium of two structures, and as certain ligands are added, they are able to bind and drive the equilibrium to the higher, more thermal stable structure of the construct. While surprising, this isn't necessarily unique. In human tissue, we have two forms of pyruvate kinase, PKM1 and PKM2. PKM1 is the form that we typically see expressed in healthy cells. However, in cancerous cells, PKM2 is expressed. What is unique about these constructs, however, is that PKM1 exists solely as an active tetramer, whereas PKM2 exists in an equilibrium of two structures, an inactive dimer and an active tetramer. Certain ligands, such as FBP and serine, can bind and drive formation of the active tetramer, wherein some molecules can bind and drive towards the inactive dimer. So with this model in mind, our next question was whether or not the disulfide bridges within the wild-type CPK are able to promote a fully active tetramer in the absence of ligands, such as we see with PKM1, while the double mutant construct actually requires ligands to allow it to assemble and become fully active, much like we see with the PKM2 construct. To answer that question, we had to turn to kinetics. The kinetics data does suggest that disruption of the disulfide bridge limits the catalytic activity of the enzyme. As you can see here, the wild type reaches a higher Vmax, or rate of production, compared to the double mutant. However, the data suggests that the affinity or the desire to bind the substrate is not affected. Again, while not necessarily surprising, as we often see decreases in activity when you create mutations on an enzyme, even if the residues that we mutate seem to be far from the active site of the enzyme. What is interesting is what happens when we add those ligands again. When we add ligands to the wild type, we see almost zero change in the enzyme's activity. This was surprising to us and something we repeated over and over again, thinking we got something wrong with our methodology. Honestly, it wasn't until last spring when I had the mental space and time to really focus on this problem that we felt confident in the data we were collecting. One thing we did notice was a significant increase in the activity in the presence of fructose 2,6 bisphosphate, and somewhat atypical activator of pyruvate kinases, and we did see the typical inhibition seen with ATP. The reason we kept thinking something was off is because pyruvate kinases from microorganisms such as malaria and TB all require some sort of allosteric activation with their ligands we tested, much like with the PKM2 model. It is not common to find a pyruvate kinase that behaves more like the human PKM1 in that it is already active in the absence of ligands and in a tetrameric form. However, I did find a study by Ditton and Coombs that demonstrated a similar trend to ours, though they were looking at cellular responses and not measuring the enzyme kinetics directly as we are here. The double mutant response, however, was very similar to other constructs in that the enzyme's activity increases in response to the typical activators. And if we look at the double mutant response compared to the wild type, not only do we see allosteric effectors increasing its activity to match the wild type, we can even exceed the wild type's activity. So what does this all mean? Ultimately, I think this project demonstrates that pyruvate kinases require assembly and that this assembly can be driven through the binding of ligands as seen with PKM2 and various microorganism constructs we have studied, or it can be driven structurally as seen with PKM1 and now potentially cryptosporidium's pyruvate kinase. The disulfide bridges may be providing structural stability that allows the enzyme to survive long term in the dormant parasite while also diminishing the need for allosteric activation once the parasite has become active and infectious. So I guess the better title for this talk would be Some Assembly Not Required. However, there does seem to be a penalty in that the presence of the bridges may also restrict conformational changes that prevent the enzyme from being highly activated, as seen in the double mutant construct. Currently, we're looking at two potential drugs that have shown promising results at inhibiting the wild type CPK, and much of this work is being done by Sydney Bates, a senior chemistry major here at Birmingham-Southern. She's planning to present her work in her senior capstone later this term and at a national chemistry conference next spring in Indianapolis. So I won't steal her thunder, but she's getting exciting results. So, what does this have to do with the importance of voice in science and my other research focus, which is in chemistry education? Simply put, the designation of neglected diseases stems from the fact that these diseases are highly understudied. As I mentioned, much of that is driven by the high cost of scientific research and the gaps in the distribution of available grant and private funding. But another aspect to this is the lack of voices, both at the benchtop and at the conference table. According to the most recent data from the NSF, white men and women make up 67% of the STEM workforce across all degree levels. At the doctorate level, however, that increases further to nearly 80%, which means that more likely than not, those who hold leadership roles and have a voice in making decisions about grant funding or which diseases to research will be white. So how does this translate to neglected diseases? Let's look at a comparison of two important diseases. In 1 year, the Cystic Fibrosis Research Foundation spent $248 million on researching new cystic fibrosis treatments, a disease that affects mostly white children, where it took the United States government nearly 10 years to match that level of spending on cryptosporidium, a disease that is the leading cause of death for children in India and Africa. To put it into perspective, about 823,000 cases of cryptosporidosis are diagnosed annually versus the 1,000 diagnosed cases of CF. In terms of patient numbers alone, cryptosporidosis poses a significantly higher public health risk than CF, but it impacts largely black and brown communities in parts of the world that are considered underdeveloped, whereas CF impacts whites in the US and Europe. Now, I'm not saying we should not be funding cystic fibrosis research. This level of funding each year has brought about important and life-changing therapeutics that have dramatically changed the outcomes for patients with CF in the last decade alone. I just think it would be equally impressive to see what this level of funding could do for kids with crypto. Also, I have many friends and family members who have struggled or lost their life to CF. I myself have a nephew with CF and was drawn into research and science in order to better understand cystic fibrosis. And there we see the problem. Just like writers tend to write what they know, scientists tend to study what they know. So if you are white and approach academic research topic through a lens of personal experience, you're more likely to be drawn to research diseases that have the greatest impact on your personal life. For me, that was cystic fibrosis, diabetes, cancer, cardiovascular disease. It is not until diseases like malaria, HIV, COVID, and Ebola directly impact whites that we see significant increases in the research funding and opportunities. And I believe that's largely because the STEM conference tables are governed by white voices. This isn't a new problem. It's one that the scientific community has recognized and studied for a while now. Most of us in the audience have heard of the STEM pipeline model and have heard complaints that the STEM pipeline is leaking, meaning we are losing potential students from STEM somewhere in their educational career, and that if we simply identify and plug the leaks, then we will eventually increase and diversify the number of STEM graduates with college degrees each year. But the problem with this model is that it suggests first that everyone can enter the pipeline, and second, if they stay in the pipeline, they will succeed equally in their careers upon graduation. But again, this concept is largely framed from a lens of those who have already made it through the pipeline, and much of the research on fixing the pipeline is focused on higher education. What I've learned in the past 3 or so years is that the pipeline model is garbage, and waiting to address leaks in the pipeline at the collegiate level is like trying to use chewing gum to plug a crack in the Hoover Dam. Our current STEM educational system is broken and tends to promote a white-dominated field where marginalized voices are lost or even blocked from entering the pipeline. In our analysis, we found that while graduation rates for Alabama schools have risen as high as 90%, the college and career readiness states reported by the Alabama State Department of Education are significantly lower at 76% across all students. The gap is wider for marginalized students, 62% for Black or African American and 69% for Hispanic. The most recent data from Alabama also showed that only 35% of students were proficient in science, meaning nearly two-thirds of our graduating high school seniors are not proficient in science. Again, an even wider gap exists for Indigenous, 25%, Black, 15%, Hispanic, 24%, and economically disadvantaged students, 21%. Further exploration into report cards for specific school systems in the state demonstrates that this is a consistent problem, especially for underserved and marginalized student populations. Simply put, our systems of how we teach science in K through 12 is currently not serving our students and should be considered an educational systems disaster for the entire state. This is especially true for students from marginalized backgrounds. First, I want to say that this is not a teacher problem. This is a systemic problem driven mainly from politicians with zero professional educational experience, and while they may have good intentions, they are not communicating enough with our teachers. We have amazing educators who are working under extremely difficult conditions and are constrained by resources, time, high student-teacher ratios, limited administrative and staff support, and rigid curriculums that are unrelatable and often delivered in a top-down manner. So, how do we address the problem that the vast majority of students coming out of Alabama schools are considered not proficient in science, math, and reading, given all of the constraints I listed will likely exist in the foreseeable future? Well, this amazing group of people, Dr. Vince Goronsky, Dr. Kelly Russell, Dr. Desiree Malonis, Roald Hazelhoff, myself, Dr. Luann Jacobs, and Dr. Mark Mead got together and talked about this problem and produced a hypothesis. If we make science more relevant, then kids will engage in science and learn. Okay, yeah, not rocket science or even novel. We've been saying that for forever. The problem, however, is that we aren't actually doing it, especially for marginalized groups. We keep teaching science, reading, and math through a largely white lens. Sure, many word problems now employ diverse names and scenarios that students from different backgrounds may relate to, and textbooks are starting to utilize images of diverse people, and all of that is great and will help. But the way we teach science is still unrelatable because they are not contextualized in a way that makes a student stop and say, oh, hey, that impacts my life. Just like researching cystic fibrosis was relatable for me. So our approach strives to ground a particular school's science curriculum with examples and challenges found in their specific communities. And if we are grounding the science curriculum within the community, then we must also bring in their social studies curriculum. And what also makes our program unique is our community of practice approach, which allows stakeholders from across the community to bring in their own expertise and experience to co-develop learning modules and service projects with teachers and administrators. Thus, the project Stemming the Tide has evolved. In this project, our goals are to bring together community agencies, educators, academic researchers, advocates, government leaders, and students to create a community of practice. A group of people with diverse skills, perspectives, and voices to share a common goal, increasing the overall health of their community while helping their students engage with and learn science. Together, this group can then collaborate to create a uniquely customized curriculum for their students that leverages service learning and the best practices in STEM education while empowering students to become agents of change in their own communities. While we believe this curriculum can be easily adapted for high school, after-school programs, and summer camps, as well as middle schools, our initial pathway has four basic steps. First, it is preparation, where educators, researchers, and community agencies are brought together in a summer workshop face-to-face to create learning modules in teams of 3. Each team is comprised of a teacher, a community partner, an academic researcher, and the distinct voice and perspective of each team member is vital to the success of the program. The teacher is the expert on the student's needs and expectations. The community partner is the expert on the relevant environmental or social challenges the community faces, while the academic researcher is the content expert and can help provide connections to resources outside of the community. While the goal of the workshop is to help co-develop and train teachers on the curriculum, it also plays a role in establishing and growing the community of practice, which is essential to the success of the program. In phase 2, sixth graders are introduced to the environmental hazards and challenges within their communities through classroom demonstrations, assemblies, and field trips hosted by the community partners. This series of events then culminates at the end of the year where all the sixth graders can attend for free the Estuary Corps Summer Camp hosted by the Alabama Coastal Foundation. This camp introduces students to ecosystems in and around Mobile, teaches them how to kayak, and trains them on the environmental monitoring techniques. In phase 3, the lessons developed in the summer workshops are implemented in the 7th grade curriculum, providing students with opportunities to learn the state standards in science and social studies while using hands-on examples from within their own communities and ecosystems that their teachers and neighbors developed. Finally, in phase 4, 8th graders will propose and implement a capstone service project in partnership with a community agency to address the challenges they were introduced to in 6th grade with the skills that they learned in 7th grade. We chose to pilot this project at Mobile County Training School in the community of Africatown for several reasons. Africatown is a community steeped in rich history and was founded by kidnapped and enslaved West Africans from the last known slave ship, the Clotilda, which was recently uncovered just a couple years ago. Africatown butts up against one of the most important watersheds of the Mobile Bay and Gulf of Mexico, and is completely isolated from other communities by industrialization and highways. According to the Neighborhood Atlas, Africatown is the most disadvantaged and underserved community in the state, if not in the entire Gulf Coast region. Ever since the enslaved captives from the Clotilda founded Africatown, their community has been repurposed, rezoned, and whittled away by the descendants of the original landowner and financial backer of the Clotilda, Timothy Mayer. As a result of decades of systemic and racist industrialization and a complete historical disregard for the environmental protection or public health by the city and state officials, the residents of Africatown have been plagued by health problems and have been fighting back in hopes of maintaining their unique community and rich heritage. Africatown is also home of the first public black high school in Mobile and one of the very few Rosenwald schools still in operation, Mobile County Training School. Founded in 1910 and accredited by the state in 1928, County now serves as the middle school for students in and around Africatown. According to the most recent data from the Alabama Department of Education, County is a Title I school comprised of nearly 100% marginalized groups. Over 90% of the student population is considered economically disadvantaged, while 22% have physical or learning disabilities, and almost 6% are considered homeless. Often, we find educational programs that leverage active learning and project-based learning, such as the one we aim to create with County, tend to target more affluent communities or families. Often these are students who are already high achieving and are funneled into accelerated programs or schools. There is a prevalent misconception in the education system that underperforming students would be unprepared and would fail if placed in such programs, and are therefore channeled into programs that focus on remedial and often ineffective educational practice. Often these students come from marginalized communities. Misconceptions like this are grounded in racist stereotypes that foster fixed mindset philosophies of how students learn and grow. And while underperforming students do require proper preparation and scaffolding to succeed in an experiential learning environment, evidence suggests that they may have the most to gain from such an approach. Additionally, their so-called underperformance is more often a result of the systemic failures to appropriately support the needs of the school and the student than it is reflective of the student's own capabilities. To date, we have hosted our first workshop, established our core community of practice group, and have begun seeing teachers implement their lessons and projects with students. It has been really exciting to see such engagement and progress this early. And I want to close by sharing an experience with you. During our breakout session of the workshop, I overheard a teacher explaining to a Mobile Baykeeper that their high school has a significantly high absentee rate. According to the Alabama State Department of Education, it ranges from 22 to 27%, compared to 3 to 9% for other schools in the area. Most administrators might look at an elevated absentee rate and blame parents or students for lack of motivation, or maybe even the school itself for not enforcing attendance. However, when the teacher said the reason was because of the significant localized flooding their area faces due to improper storm drainage, trapping students in their home, the problem was clear. This is not a result of poor motivation or enforcement. This is an example of racially driven environmental injustice, and just one of the many that the community of Africatown endures. After hearing this problem, the Baykeeper exclaimed, "Hey, I can come to your class and show students how water tables work and how we can predict flooding." Immediately, the conversation took off as the group expanded on the idea, brainstorming how students could first learn about water tables through the Baykeeper's demonstrations, then take mapping data collected from another group through drones and construct water tables for their own neighborhoods to predict where and why flooding might be occurring at their homes. Later, the Baykeeper admitted, "I never get around to visiting County. I wonder why it's always last on my list." The reason was clear to me. They never had a chance to sit in the same room and talk. Once teams were told to identify state learning outcomes and outline learning activities, the curriculum nearly built itself. I was shocked when, in less than an hour, teams had drafted innovative plans with learning goals, assessments, implementation strategies, and spin off projects. The energy of the room was contagious, a stark contrast to the typical workshops where educators are talked at. Why was our approach different? I think it's because we entrusted that each participant had value and voice, and that all had a common goal to ensure that students are engaged and learning. Imagine what diverse voices like this can do for science when seated at the conference table together. I want to take a moment to thank our Africatown community partners who have all jumped at the opportunity to engage with the students and faculty at County, without whom this project would not work. I also have to thank the teachers at County. Their love for their students, school, and community is unparalleled. I've never seen a group of teachers so dedicated and driven to help their students succeed. And of course, I have to thank the Gulf Research Program from the National Academies of Sciences, Engineering, and Medicine, who have invested heavily in our research and continue to find new ways to support this project. Finally, I have to thank my students, whose insight and hard work our research would never evolve or grow. Nearly everyone pictured here has worked on the pyruvate kinase project in some way, and all of them have influenced my voice as a mentor and an academic researcher. If you're looking for more opportunities to learn about stemming the tide and about the history and importance of preserving the voice of Africatown, Dr. Jacobs has curated a book display in the library of recommended readings. This Friday, Netflix will be streaming the documentary Descendant, which documents the history of the kidnapped and enslaved West Africans who founded Africatown, their descendants, and the history of the town and the discovery of the Clotilda in 2019. And finally, on November 12th, the Stemming the Tide leadership team will host a discussion panel on the project during Common Hour. This is also a Lectures and Arts event. I hope to see you all back for that discussion. And finally, to my students in the audience, hone your voice and use it to craft words of compassion and to elevate the voices of others. And always remember, the only thing more painful than the weaponization of words to shame and silence is the complacency that follow in their wake. Thank you, and I'm happy to answer any questions.